Hello everyone. In this lecture, I will be covering structure and function of prokaryotes. First, we will be talking about the structure of bacteria, then function, classification, and reproduction. So, in between the lecture, there will be slides like these that will pop up with questions. I would like you to pause on these slides, answer these questions to the best of your knowledge based on the videos you have seen, the labs you have done, and the case studies you have gone over in the class and the lectures. So there is no right or wrong answer. This is just to gauge your prior knowledge. Here's another question. And finally, here's the third question. So let's start talking about the structure of the bacteria. First of all, I'm going to start off with extremophiles. That means these bacteria are found in harsh conditions. They are found in places that are too acidic, salty, cold, or extremely hot. They are very prolific. That means they're found everywhere. Handful of soil can contain more bacteria than number of people that have ever lived on this planet. Here's another interesting fact for you guys. You exchange 250 species during a kiss. And when you use or share those headphones, you increase your bacteria population by 700 times. These are the three shapes of bacteria that we have constantly talked and looked at in the class. The first one is your spherical or cocci. The second is your bacillus or rod-shaped bacteria. And finally, the spirillum. This is the structure of bacteria. This is how bacteria looks like. There are different components and internal organelles that are not membrane bound. I will be talking about each of those components one by one in these slides. Let's start off with the cell wall. The cell wall of the eukaryotic cell is made up of cellulose or chitin, whereas the bacterial cell wall contains peptidoglycan. Now, peptidoglycan is a polysaccharide. Along with peptidoglycan, the cell wall contains cross-linked chains of polypeptides. You have been working on gram differential staining. That means that's the first type of staining that helps you classify bacteria into gram-positive and gram-negative bacteria. I would like you to look at this picture and focus on the left side first. On the left side, you see this is the cell wall of bacteria. This is the peptidoglycan layer in which when you add crystal violet, the crystal violet stain gets trapped in between this peptidoglycan layer, therefore giving this blue or purple color to your bacteria, indicating that bacteria is gram-positive. On the other side, the gram-negative bacteria is made up of two layers of lipopolysaccharides, and then in between is the peptidoglycan layer. Now, when you add crystal violet stain, it masks the lipopolysaccharide layer but gets rinsed off. But when you add saffron in, saffron goes in and gets trapped in the peptidoglycan. And therefore, you see the red coloration of bacteria for gram-negative. Now, antibiotics we know can kill bacteria. And unfortunately, gram-negative bacteria is the one that is pathogenic. And the cell wall of the antibiotics, they destroy the cell wall of the bacteria, therefore preventing it from growing and damaging the cells. Unfortunately, again, gram-negative bacteria, the cell is the one that gets mutated more frequently and therefore becomes antibiotic resistant. A polysaccharide layer or protein layer called a capsule covers many prokaryotes. Now, I would like you to focus on the long threads or the long strands that are coming out of the bacteria in this picture. They are called fimbriae. Fimbriae are used by bacteria to attach to the surfaces, whereas the smaller fringes that are coming out, or the threads that are coming out, are called pili. They are used for reproduction, which is the sexual reproduction, so-called sexual reproduction in bacteria, conjugation. Motility is another way that you can classify bacteria. Bacteria can be either motile or non-motile. Many bacteria exhibit taxis. That means its ability to move towards or away from stimulus. As the name suggests, chemotaxis means movement toward or away from chemical stimulus. And a bacteria can move up to 50 body lengths. Pretty impressive, right? I would like you to watch this video to see the motility of bacteria. Pause the slide, click on the link, watch the video. Flagellum is another important organelle of bacteria that helps in motility. The flagellum is made up of three major parts: a filament, the hook, and the motor. The internal organization of DNA of bacteria is very simple. All the organelles are laid out in the cytoplasm, and they are without the membranes. So therefore, they are not membrane-bound organelles. All the DNA gets concentrated in one region of the cell, and that region is called nucleoid. Some species of bacteria contains a smaller ring of DNA called plasmids. Prokaryotic ribosomes are small and slightly different, and they're distributed all over the cytoplasm. Now, ribosomes, remember, help in assembly of proteins, and proteins are required for the growth of bacteria. So it would be right to make the antibiotic that's going to target ribosomes. So erythromycin targets the ribosomes, destroys them, therefore prevent the assembly of proteins. And as a result, the bacteria do not have the proteins to grow and dies. Reproduction and adaptation. Prokaryotic reproduction is quick. Binary fission occurs every 20 minutes on average. That means the mother cell divides up into two identical daughter cells. Rapid reproduction brings in considerable genetic variations. Now, there are three factors that contribute to this genetic variation: rapid reproduction, mutations, and genetic recombinations. Prokaryotes reproduce by binary fission, as I said before, and because even though binary fission produces identical cells, the rapid reproduction causes mutations. For example, on average, there are 9 million E. coli mutations per day per human host. Bacteria, as mentioned before, can survive in harsh conditions, and what enables them to survive is what's called endospores. So when bacteria, the conditions are not optimum, the bacteria gets distressed. Under that distress, bacteria will produce what's called endospores. If I have to give you an analogy, endospores are like the bacteria lays eggs, and those eggs can survive in the harshest conditions until you place them in autoclave and destroy them completely under extreme temperature and pressure. Researchers have revived 250 million year old bacteria from endospores that's found in the salt crystals. So what's genetic recombination? As the word suggests, it's recombining of two different bacteria. That means the foreign bacteria comes into the original DNA of the bacteria and combines with it. This recombination takes place by three different processes: transformation, transduction, and conjugation. Movement of genes among individuals from different species is called horizontal gene transfer. Now I would like you to pause at this slide, click on each of these links, watch these videos, and then come back and continue on with the lecture. So transformation and transduction, as you have already seen the video, so I'm going to go through this quickly. Transformation is incorporation of foreign DNA in bacteria. Transduction is movement of genes between bacteria by bacteriophages. And finally, conjugation is sort of like the sexual reproduction in bacteria, where genetic material is transferred. So one bacteria becomes the donor attaches the recipient by a pilus and transfers the DNA. A piece of DNA called the F factor is required for the production of pili. The F factor as a plasmid functions as a DNA donor during conjugation, and it's a one-way delivery. It transfers during conjugation and the recombinant DNA is formed in the recipient cell. Here's a question: why you should resist hand sanitizer? Maybe. So pause and try to think about the answer. Our plasmids carry genes for antibiotic resistance. Remember, our plasmid is in the recipient cell of conjugation. Through natural selection, the fraction of bacteria with genes for resistance increases in a population exposed to antibiotics. So what would be the outcome? Diverse nutritional and metabolic adaptations have evolved in prokaryotes. So we have autotrophs. They require carbon dioxide. That means they can make their own food. Photoautotrophs, they obtain energy from light. Chemoautotrophs obtain energy from chemicals. Chemoheterotrophs, they require organic nutrients to make organic compounds, energy from sugar ingestion. Chemolithoheterotrophs have been found miles under Earth. Now try to define photoheterotrophs. So what's the role of oxygen in metabolism? Prokaryotes can be obligate aerobes. Aerobes means they require oxygen for cellular respiration. Obligate anaerobes are poisoned by oxygen and use fermentation or anaerobic respiration. And finally, the facultative anaerobes can survive with or without oxygen. Here are a couple more questions for you to answer. So please pause, answer, and then move further. So nitrogen fixation. Some prokaryotes convert atmospheric nitrogen to ammonia. This is a hint to your previous question. Cooperation between prokaryotes allows them to use environmental resources they could not use as individual cells. In the cyanobacterium Anabaena, that's the name of the bacteria, photosynthetic cells and nitrogen-fixing cells called heterocysts exchange metabolic products. Molecular systematics is illuminating prokaryotic phylogeny. That means how the whole kingdom, archaea, is divided up. And here's a picture. You can pause and study this. Here's another question for you. Based on the previous one, answer this question. Archaea share certain traits with bacteria and other traits with eukaryotes. Archaea share certain it's a repetition. So comparison of the three domains of life. You can pause on this slide and look at the chart. We've talked about extremophiles, halophiles, and thermophiles. Methanogens live in swamps and marshes. And finally, we're getting to the conclusion of this PowerPoint. So hopefully you've enjoyed this lecture. And if you have any questions, you may come to my room at any time. These definitions I've already talked about, and these are just few repeated vocabulary